did very well. So I'm proud about this. So it looks like you're very attentive in the class, and yeah, so it's very good. So I'm going to, so most of the questions, you know, I think you know that you got from the slides. As long as you pay attention to the class, and you know, you don't even have to take notes, you can actually get through these exams, you know, pretty fairly well. So I'm proud about that. And about the exam too, you know, we had a request that this be postponed by 1 or 2 days. So if I can get the oaths from her today, if all of you agree with that, then we'll postpone the exam by 1 or 2 days, and then Scott Sieg, Dr. Sieg was not here. He might use that slot for giving his lecture, like the replacement time. So that's the whole idea, but it really depends on you know, what you all agree with. Okay, so
yeah, guys, if you didn't vote yet, just it went on an email and a remind yesterday. Ouch.
okay. All right. Okay. So today we are going to, you know, so the second portion of this body is host. As you can see, it's more related to the, you know, the pathogens or the microbial side. Okay, so you are looking at bacteria, you know, fungi and viruses in detail. So that's what we're looking at. So today we are going to be looking at the fungi, fungal diseases in the context of immunity towards the fungi. Okay? So this is the concept map. First of all, we are going to look at some of the fungal diseases and types. It's mostly, you know, a list of a lot of fungi causing different manifestations. And then, as you will see, most of these are caused by, you know, Candida and some other commensal microorganisms or fungi. So we will, you know, look at the details of the Candida, virulence factors of Candida, and the host immune responses to Candida. Okay? And then we'll also touch upon a little bit about the diagnosis methods and anti-fungal treatments currently in use, and then you know, that will end the lecture. So human fungal pathogens, first of all, are divided into two. So the first are the primary fungal pathogens, and the second one is opportunistic fungal pathogens. Okay, so we'll look at the details about what these are and how they are you know, divided into. Before that, you know, there's a technical term for fungal diseases. What is that? It's in this slide. Mycosis, yeah. So myco is a Greek word. It's derived from a Greek word where myco means mushroom or fungus. So, so mycosis are fungal diseases. So let's look into the details of the primary fungal pathogens, and these distinct characteristics are, you know, important to know. Like, there's not too much about, you know, remembering the types of diseases they're producing, but you know, these the types of fungal pathogens and how they are different is important to remember. So primary fungal pathogens are capable of initiating infection in a normal, apparently immunocompetent host. Okay, so, and it's able to colonize the host. It can find the suitable microenvironmental niche with sufficient nutritional substrate. So so the fundamental thing that you have to remember is that even immunocompetent hosts can get these infections by these primary fungal pathogens. Okay, so because it has certain features, virulence factors that enable them to attack the host, and and these virulence factors enable them to avoid or subvert the normal host defense mechanisms, so both innate and adaptive. So which is why they're able to multiply within these microenvironmental niche, and that's why they are primary fungal pathogens, or they are there primarily to cause the disease, okay? Some of the examples are ascomycetes fungi, such as blastomyces dermatitis, coccidioides immitis, histoplasma capsulatum, and paracoccidioides brasiliensis. So these cause big fungal diseases, and these are primary fungal, some of the key examples of primary fungal pathogens that you should remember. And opportunistic fungal pathogens we touched upon a little bit when we were looking at the immune system in our last class. So these are present as normal part of the or the normal commensal flora, and which is present endogenously. So it's or in the environment as exogenous source of these opportunistic fungal pathogens. Normally, they do not cause infections. Most of the time they don't. Okay? But when there is a disruption in the protective barriers of your immune system, like in skin or in other mucosa, then they become opportunistically pathogenic. Okay, so that's basically when there is an immunocompromise in the host. Okay, so only then they're able to penetrate, colonize, and reproduce in the host. And some of the key examples of opportunistic fungal pathogens are Candida species, Cryptococcus neoformans, and Aspergillus species. Okay, so these are the key examples. So Histoplasma capsulatum is one of the biggest primary fungal pathogen that you will see often in a lot of the diseases in the subsequent slides. And Candida albicans is the key example for the opportunistic fungal pathogen. So I just mentioned in one of the slides that most of the primary fungal pathogens are ascomycetes fungi. Does anyone know what what ascomycetes and basidiomycetes are? These are like based on their morphology or structure. These are two types of fungi. And they're categorized into ascomycetes and basidiomycetes. Does anyone know? It's not in the slide. Okay, so look up on these. You might get a question on ascomycetes and basidiomycetes. Not like detailed question, but something relevant to ascomycetes and basidiomycetes. Okay, so let's look at the fungal diseases. The most common ones are abdominal infections and respiratory infections. So let's get started with the respiratory infections. So the first thing is oropharyngeal, oral infections caused by fungi, and the species that are causing oropharyngeal infections are Candida, Cryptococcus. These two are opportunistic fungi. And even Histoplasma capsulatum and blastomyces and paracoccidioides can also, cause oropharyngeal infections. Okay, so wherever bacteria can infect and cause infections, fungi can also do that. Okay, so some can be opportunistic, some can be primarily pathogenic fungus. And they also cause sinusitis, sinus infections, and most common one species of fungus that causes sinusitis is Aspergillus species, and followed by mucormycetes and Fusarium and some other molds. Okay, so sinus infections can be caused by these. And laryngeal infections in the larynx and esophagus can also be caused by the primary pathogen Histoplasma capsulatum, but mostly esophageal infection like oropharyngeal is caused by Candida, which is an opportunistic fungus. So, bacteria can cause otitis, the ear infection, external otitis. Aspergillus niger, Candida species can also cause ear infections. So this is a pictorial representation of how Aspergillus looks under the microscope. And they also cause eye infections. A lot of us might have come across. Use of contact lenses can cause fungal infections in the ear, and it's mostly caused by Candida and also others. It can cause endophthalmitis, keratitis, and lacrimal sac infections and infection in the short channel near the inner ear corner of the eyelid. So it's called canaliculitis. Okay, so and mostly it's caused by again Candida albicus and Aspergillus niger. So, but endophthalmitis and some corneal infections can also be caused by some primary pathogens such as Histoplasma capsulatum and opportunistic pathogen Cryptococcus pneumonitis.
Did you say that the contact lenses can cause an infection in the ear?
No, in the eye.
Oh, okay.
Yeah. Yeah.
It's like traveling through.
So, again, more respiratory infections, pulmonary and bronchial infections causing bronchitis and pneumonia, and fungus ball. So it's a, fungus ball is a very typical manifestation caused just by fungi. We're going to be looking at it. These are again mostly caused by opportunistic infections by Aspergillus, Cryptococcus, and also mucormycetes. Okay, so fungus ball is caused mainly by Aspergillus species, and it's actually a ball of hyphae, mixture of mucus with a mixture of mucus and cellular debris, the immune cells, and it colonizes the parenchymal or the lung cavity, and that's called fungus ball, and it can cause pneumonia, obstruction of oxygen flow. And so this doesn't happen normally, but when it only happens in people who are hospitalized and undergoing some steroid treatments or with extremes of age. Okay, so this is an X-ray of a fungus ball in the parenchyma of a lung of a patient, and caused by Aspergillus niger, which is again an opportunistic infection. So, and genitourinary tract infections and prostate infections are also caused by some of the opportunistic fungi, vaginal and prostate infections, and Candida and Saccharomyces cerevisiae yeast infections are commonly known, the most common infections, and they can cause also some prostate infections. And intra-abdominal infections again are caused by fungi. In the peritonitis is an inflammation in the membrane that's covering all the abdominal organs, so that's called peritoneum, and that can get infected and inflamed. That's called peritonitis, and that's caused by Candida, because Candida is present in the oral cavity of normal humans, and when they take a lot of antibiotics, they can translocate into the gut and the peritoneum, causing this peritoneal infection or peritonitis. Okay, so it's rather uncommon, but they have isolated Candida from such infections, and the visceral abscesses can also be subsequently result due to these gut infection. So in extreme cases, they can also cause cardiovascular infections in endocardium and the pericardium, where Candida and Aspergillus species are known to cause these infections. So it has also been reported that fungi can cause infections in the central nervous system. So mostly people believe that meningitis is caused by bacteria. So meningium is the membrane covering the spinal cord. It's like a 3-layered membrane covering the spinal cord and the brain, and crossing the blood-brain barrier. Some bacteria can cause bacterial meningitis, but now it's known even fungi can cause meningitis, and most common species are Cryptococcus, Aspergillus, and Candida, and that happens again with during hospitalization and corticosteroid intake and antibiotics intake, and they can also lead to brain abscesses. Yeah, so this is one of the most common, infections, fungal infections caused on the skin, and mostly it's caused by not Candida, Malassezia, and that is again another opportunistic pathogen. It lives on the skin all the time in most individuals, like 80% of the individuals, but it causes skin and it can invade the soft tissues and can cause superficial and cutaneous infections. Okay, so Malassezia species and dermatophytes and some molds are common culprits causing these skin infections. They can also lead to infections post wounds or surgical or traumatic episodes. And that's mostly caused by Trichosporum and Rhodotorula. So these are not very common, as you can see. I've underlined this, but where wounds are infected by these fungi and happens during hospitalization. And some of these skin infection can become hematogenous, which means that can be spreading to the entire body through the blood and then have other consequences. So Malassezia, as you all know, some of us have come across dandruff, and that's caused by a fungus called Malassezia species, and that's normally a monophylogenetic genus of Bacillomyces fungi, and it's found on the skin of 7 billion humans. So a lot of humans do have this normally, but it causes dandruff in some people, and some of us are aware of this. And it's not only in immunocompromised hosts. Malassezia can also cause stomach infections, but fairly immunocompetent people can get dandruff, but it can be easily treated and removed. But maybe the pH differences in the hair can lead to the development of dandruff or the invasion by Malassezia in the scalp. So there are 14 currently recognized species of Malassezia, 8 of which have been associated with humans, and 4 of these are commonly involved with the dandruff infection. So they also can cause osteomyelitis, which is bone and joint infections, and arthritis. That's arthritis is partly immune inflammation and partly fungal infection. Mostly it's bacterial infection, but fungi can also cause arthritis and inflammation in the joints. Even prosthetic joints can get infected, and mostly it's caused by Candida. Okay, so this is the another picture of Histoplasma capsulatum. In the exam, you might have some pictures to recognize which fungi are which. So, pay attention to these structures in mycelium and the hyphal structures of these fungi. So the prosthetic joint infection can also lead to hematogenous dissemination or bloodborne infection, and most of the fungi can do this, and but under the conditions of immunocompromised hosts. So as you can see, a lot of the times, 50 to 60% of the times, the fungal infections are opportunistic or caused by opportunistic fungi. These are the predisposing features of these opportunistic mycosis. First is antimicrobial agents like antibiotics that can kill off some of the good bacteria, can let the fungi invade the system. So if the number of the antimicrobial agents and the duration is pretty high and long, then it can definitely become a predisposing factor for opportunistic mycosis. And corticosteroids, obviously these are immunosuppressants. Long exposure to corticosteroids can also, make people susceptible to fungal infections. And chemotherapy and where you have solid organ malignancy, can also kill a lot of immune cells and can suppress your immune system and thus can make the patients become more susceptible to these infections. And previous colonization, like I said, 80% of the people, 60% of the people have these Candida or Aspergillus in their normal body or Malassezia, for example. But if the previous colonization levels are high to begin with, then it is also a predisposing factor. And indwelling catheters and total parenteral nutrition, can also expose your body, internal organs of your body to the external plastic tubes, and these can have the fungi living on them, and that is a predisposing factor. And generally neutropenia, or the WBC count less than 500 per millimeter cube, is considered to be a very big factor for opportunistic mycosis, and this happens during chemotherapy and corticosteroid therapy, and also during immunosuppressive infections such as HIV infections. Okay, so these can lead to opportunistic mycosis. And as you can imagine, the external barriers and epithelial cells are severely disrupted during surgery and burns, and that happens to be a predisposing factor for fungal infection. And assisted ventilation, again, like parenteral nutrition, can expose your internal organs to the tubes and external plastics that can harbor fungi. Okay? Again, hospitalization and dialysis patients have a lot more exposure to stuff and fungi, so that's a predisposing factor. And in developing countries, malnutrition and which is leading to immunosuppression, or a lack of proper nutrition also leads to fungal infections. And extremes of age and immunosuppressive infections such as HIV that causes neutropenia, again leads to fungal infections, or predisposition to opportunistic mycosis. So as you can see, immunosuppression and Candida species, they are coming, we hear these two again and again. So we look at some of the details of how Candida causes the infection and how immune system is developing antimicrobial response to Candida. So this is, I think this slide we already saw. Oral candidiasis are, 4 types of candidiasis. So the first is pseudomembranous, second type is acute or chronic atrophic candida, and the third type is angular cheilitis, and the fourth type is chronic hyperplastic candidiasis. Okay, so the first one is the most common one, where you can see white blobs on the mucous membrane in the buccal cavity, and erythematous can be it's like a reddishness, and sometimes people wearing dentures can have erythematous candida infection. Like, it's not as obvious as white patches, but then they have reddishness and swelling accompanied by burning and pain, and that's erythematous candidiasis. And angular cheilitis is an infection caused by Candida on the cracks and crusts in the commissure area where the two lips join. In developing countries, a lot of kids have angular cheilitis Candida because of malnutrition, and that usually is seen in the commissure area, where like these are cracks and crusts and white patches on these commissure area. And chronic hyperplastic candidiasis is, people have this candidiasis come back again and again, and that's because of antifungal resistance, and that is usually characterized by epithelial cell thickening, and it's very similar to leukoplegia, which means enlargement of leukocytes or infiltration of leukocytes under the epithelium, which will cause swelling, and that never goes off, and that's why it's a chronic disease. And a lot of aged individuals have chronic hyperplastic candidiasis. And all of these are either caused by loss of T cell functions, Th17 cell functions. We have some slides for that, and also if you don't have proper immune regulation, that also leads to excessive immune responses and accumulation of inflammatory cells causing chronic hyperplastic candidiasis.
There's a question.
Yeah.
In the slide before, the bottom picture, is that chronic hypoplastic then?
So this is a picture for erythematous candidiasis showing some reddishness in the gingiva, but leukoplakia can also happen. This is like something associated with dentures or similar to dentures, but hyperplastic candidiasis can happen anywhere in the buccal cavity. So the most common causative for thrush is Candida albicans, and it's a resident fungus in 60% of healthy individuals. In one line, host resistance is mediated by nitric oxide radicals and myeloperoxidase activity by neutrophils. And macrophages can also phagocytose these fungi, and adaptive immune response is mediated by a predominant Th17 response. Okay, so it's the most common fungal opportunistic infection. Most of us don't have an infection, but only people with AIDS, APSID. APSID. These patients have mutations in AIRE, the central tolerance defect patients we saw last time, and even people with diabetes, excessive sugar, giving excessive nutritional source for these fungi can also be a predisposing factor for these oral candidiasis. And I mentioned when there is an absence of immune regulation, there is candidiasis in IPEX patients who also lack regulatory T cells. Some of them have candida infections on the skin. So, like we mentioned, this is like a review slide. Opportunistic fungal pathogens. These are the examples, and these exist as benign colonizers or environmental saprobes, and only cause serious infection when there is a breakdown of host defense. And let's look at the pathogenic factors of Candida. So they colonize to begin with the oral or the gastrointestinal mucosa, and they reach the bloodstream through gastrointestinal translocation via contaminated catheters, and they have capability to interact with host defense, and they can also invade intravascular compartment, and then go and cause infections in deep tissues, causing liver, spleen, kidneys, heart, and brain. Okay, so that means they have very potent virulence factors once they start behaving like a pathogen. Okay, so what are these virulence factors? So their virulence factors, like many of the fungi, like it's an example of one of the fungi. This will be common to many other fungi. So the virulence factors include the ability to adhere to inanimate surfaces and tissues. Okay, so they have special factors that leads them to adhere to inanimate surfaces, like dentures or catheters, and so when you're exposed to them, you're easily infected. The other important thing is they have dimorphism. That means they can exist in two forms. One is yeast, and the other is the hyphal form. When it's in yeast form, it's non-invasive. When it's in hyphal form, it is invasive. Okay, so as long as it's yeast, we don't have any infection, and its ability to transform into hyphal form lets it to invade the internal tissues, and that is one of the virulence factors. And cell surface hydrophobicity, proteinase secretion to destroy some of the host factors, and phenotype switching. all these are other virulence factors where Candida used to invade the host. So adherence is achieved by a number of attractive forces. The first is ligand-receptor interaction. So they have certain ligands binding to some of our receptors on the host cells, and non-specific interactions such as electrostatic and van der Waals forces which enables them to adhere to the host. So what are these factors? So they have glycosylation of manoproteins, so they have some glycoproteins or sugar-attached proteins to the cell surface, and that makes them really hydrophobic, and exclusion of water can make them attach to the epithelial cell much better than the other fungi. And they also express certain other glycoproteins like ALS, which is a protein-like sequence, and epithelial adhesin. So these are like really adhesives binding to the epithelial cells. And this is like a non-specific sense of touch, thigmotropism. Concept where Candida albicans can grow through the grooves and through the pores and may aid in infiltration of epithelial cells. So wherever you have pores and grooves, like for example in dentures, they can like really grow in high levels, and then your epithelial surfaces are exposed to high loads of these fungi, which leads to invasion. So this process is called thigmotropism, by which they can attach to the epithelial cells. So this is van der Waals forces. These some of you might have had chemistry or physics where you might have read about van der Waals forces. So this is also how Candida invades dentures. It's basically increase in the surface area leads to the shift of the forces, and the positives and the negatives of the adjacent forces to come close to each other, and that makes them attach. It's a weak force, but still Candida has these nanostructures, like for example here in gecko, it has some really curls at the edges of the feet, the gecko. Even it can attach to the glass because of these structures that leads to shift in charges, and lets them bind to any surface they want. Okay, so these are nanostructures under the side of under the legs of these webbed feet of the gecko. So similarly, Candida have these nanostructures that enables them to bind to the epithelial surfaces. So the other feature is the expression of proteinases. Proteinases are the enzymes or proteins that can cleave other proteins. So they have aspartyl proteinases that can hydrolyze proteins involved in host defenses. So, for example, if the host is producing an antimicrobial peptide, so they have aspartyl proteinases to cleave those and then bypass the innate antimicrobial peptide production by host immune system. Okay, so they also have phospholipases that can cleave a lot of lipids produced by the host, and thus they can lead to damages in the epithelial cell and cause tissue invasion. So the other one is the phenotype switching or the dimorphism, where they can convert from yeast to hyphae and hyphae to yeast, and that is usually regulated by the pH of the mouth. So if the pH is too it's too basic, it can lead to hyphae formation, and so if you have a lot of sugars, if you eat a lot of sugars, that can lead to pH changes, as you all know. and that can be a predisposing factor for the yeast to convert into hyphae. And that ability is also is a big factor for them to cause infection. So that's this. So these are yeast structures along with some hyphae. This is called pseudo hyphae because these are sort of elongated yeast. It's not real hyphae. But here you can see this yeast has germinated into hyphal structure, the elongated structure, and this is invasive part. Okay, so if it's in the yeast part, if it's like in the yeast form, it's not really able to invade. Okay, so once they develop into hyphae, then they start producing these proteinases and lipases and lets them invade, and also they can grow along the grooves and all that with these hyphal structures. So these leads them to evade the immune system and also to invade the host. Okay. So this is again a review question. What distinguishes a primary pathogen from an opportunistic pathogen? Primary pathogens are capable of initiating infection in normal people. Opportunistic only when there is an immunocompromise or when there is a shift to pH. There is some disruption in the local mucosa. Yeah, so we looked at the dimorphism and the virulence factors, and these are some of the review slides. I want you to remember. This might be some of the features that we will include questions from. Okay, so now from this, let's look at the host side, the immunity to fungal pathogens. So this is a general slide where a host immune system is mounting an immune response to microbial pathogens. Okay, so from here, let's get into the fungi because it's like a review slide from some of the immunology classes that you had before. So a microbial pathogen invades. They express what's called PAMs, or pathogen-associated molecular patterns. And these patterns somehow activate through different mechanisms because these molecular patterns are very specific to certain different classes of bacteria and fungi. So these PAMs activate different classes of leukocytes and humoral components, which is like antibody components. So leukocytes that are involved can induce antimicrobial peptides, taking care of the innate removal of bacteria or fungi, or phagocytosis. Okay, so your macrophages, neutrophils come and can phagocytose the bacteria or fungi. They can also produce some of the cytokines required for recruiting other cells to the site of infection. So these are some of the key things that's happening once there is a microbial pathogen attacking the immune host, okay? Or fully immunocompetent host. Okay, so and these cytokines can bring in more phagocytes to increase the microbicidal activity. They can bring more cells, and it can also lead to bone marrow producing more immune cells, and it gives some signals to the central nervous system to let the body know that it has to respond primarily to this to attack this pathogen. So that's what is coming out as fever, and that's also because liver produce a lot of acute phase proteins that also does a lot of things to clear the infection from the blood if it comes to the blood. Okay, so this is somewhat what is happening to almost all infections. So what's different about fungal? Like there are some features that are different here, and that's how it is for viruses and bacteria. So those are the distinct things that are happening here. Like how the first line of recognition happens is what is different with these microbes. So, like I said, the fungi have glycoproteins. That's very exclusive to a lot of fungi. Enzymosands, beta-glucans, and phospholipomanins. And these as PAMs or pathogen-associated molecular patterns that are associated with fungi. Okay, so and it's not in bacteria, it's not in virus, but these are exclusive to fungi. Okay, so and the immune response or the immune cells have specific receptors to respond to these guys. Okay, so in Candida albicans, these PAMs are present, and the immune cells have dectin-1, 2, and TLR2, and these are the pathogen recognition receptors that respond to these glycoproteins, zymosands, and phospholipomannins. Okay, so these are present in many innate immune cells. In the first stage, right here, the PRRs, the pathogen recognition receptors that respond to the glycoproteins present on the fungi. Okay, so here is a fungi. So if you look at the enlarged picture, these are the structures of beta glucans, which is the one of the primary PAMs in the fungi, and these are PRRs, the dectin receptor and toll-like receptor. So it leads to the activation of certain signals, activating NF-kappa B and MAP kinase as well, leading to the production of these types of cytokines. So which is TNF alpha, IL-12, IL-23, and these are the ones that are going to recruit Candida specific or fungal specific adaptive immune cells. Okay, so this pathogen recognition receptor signal is very specific to the beta glucans present in Candida. So we saw this, the components of the innate and the adaptive immune system, and macrophages, neutrophils, T cells, and dendritic cells. All of these have these pathogen recognition receptors that recognize fungi. So we looked at this before. The two types of T cells are CD4 T cells and CD8 T cells. It is believed that in mouse, even CD8 T cells can be involved in antifungal immune responses, but predominantly adaptive immune response is mediated by CD4 T cells, especially T helper type 17 cells, T17 cells. Okay, so that's in a nutshell. In mouse, you have a fungus. It can be phagocytosed and killed directly in a macrophage or neutrophils. It can also produce cytokines and chemokines to recruit other cells, and it can also activate immune responses. So in this case, in mouse, the immune system activates Th17, Th1, and CD8 cytotoxic lymphocytes. In humans, it only activates Th17 and Th1 cells. Okay, there are some cells that can make interferon gamma that is also known to have antifungal activity, but Th17 cells and recruitment of IL-17 producing cells is more important in antifungal killing, antifungal responses. Okay, so in mouse, they have shown both mucosal and systemic immunity. In humans, it's basically just the mucosal immunity mediated by Th17 cells. Okay, so the dectin can activate NF-kappa B in dendritic cell. It's IL-10 production, and you have to have immunomodulation in the context of infection. And IL-10, as you know, is an immunomodulatory cytokine that prevents excessive immune responses to fungi. So that is also activated by these beta-glucan binding to dectin receptor. Okay, and TLR1 and 2, it's a heterodimer. That can also lead to MYD88, an adapter activation, leading to NF-kappa B activation, and this is IL-12 that induces Th1 responses, so that's how it does the activation of Th1 responses. So what they have not shown here is this NF-kappa B can also lead to the production of IL-23 in some of the antigen-presenting cells, and that leads leads to the induction of Th17 responses in the epithelium. Maybe like we have that in the next slide. Okay, so here's what is happening at the point of contact. Here the fungus, and it binds to the PRR cluster, activating NF-kappa B and MAP kinase and leading to the production of cytokines. And this happens in a macrophage here. So this is in the context of beta-glucan binding to dectin-1. So yeah, here you can see it's not only inducing the development or the production of IL-12, it also leads to the production of the dectin signaling, dectin-1 and 2, the beta-glucan and the alpha-mannan together. So this is beta-glucan, alpha-mannan together, for example in Candida albicans, can combine together to a synergistic pathway, and that leads to the expression of IL-1 beta, IL-23, and these are the key cytokines that can induce Th17 response. Okay, so you need both dectin-1 and dectin-2 to be activated in a dendritic cell, whereas just dectin-1 alone can lead to only the expression of IL-12. Okay, so because we're going to look at the Th17 responses in detail, here you should remember both the PRRs, dectin-1 and 2, are required for IL-23, IL-1 beta that can induce Th17 differentiation in T cells. So this is what is happening in the context of aspergillus in a lung infection or allergy caused by molds, where you have lung epithelium, and the immediate response is generated by tissue-resident macrophages in dendritic cells, like here. Okay, so it can bind to dectin-1, 2, and all that. So that's what is happening here, and they produce cytokines and chemokines, and they recruit the first wave of cells, and the first one being the neutrophils. Okay, and the neutrophils are primarily antifungal due to the fact they can produce myeloperoxidase and nitric oxide radicals that can directly kill these fungi. Okay, so that's the first wave of recruited cell that happens so rapidly within few hours. Okay, so they can take care of the fungi right there. But these chemokines and cytokines also activate the second wave of recruited cell to make sure that you don't have a reinfection. And the second wave are monocyte-derived dendritic cell, plasmacytoid dendritic cells, and T cells as well. Okay, so and these actually aid in fungus-specific T cell priming, so you have memory cell responses. Okay, so these are essentially recruited because of the cytokines and chemokines produced by the tissue-resident macrophages. For example, in lung tissue, you have alveolar macrophages doing this. In different tissue, it's different. Okay, so this is happening because of, again, TLR2 and dectin signaling happening within these cells. So what happens once the T cells come in? This is the second wave of recruited cell. What's happening is, what's relevant to antifungal response is Th17. Like I said, they are named so because they secrete IL-17, and it plays an important role in cell-mediated immunity and important defense against fungi. So what do they do once they come in? So here is an example. This is a gut example, but this is the same. Similar things happen in the oral cavity as well. So here you have epithelial cell layer producing these antimicrobial peptides, lipocalin, calprotectin, and beta defensins, and REC3 gamma. These are the antimicrobial peptides that are produced as a part of the innate defense against fungi. Okay. But you know that these Candida, for example, can have proteinases and lipase to cleave these, and that's why they actually get into the system. Okay? So once they get into the system, Now through PRRs and PAM interaction, they activate the local dendritic cells and macrophages and that produce IL-23, and IL-23 is a Th17 inducing factor or IL-17 producing cell inducing factor. So CD4 T cells expressing IL-17 is happening right here, and one of the main functions of IL-17 is to produce, to activate chemokine production and metalloproteinases in local cells. Okay, so they form another innate layer right here, but then they also recruit more neutrophils. Okay, so that those neutrophils can again lead to the destruction of fungi. And these cells, once they do take part in this innate cell activation, they go and form memory cells, and you can find, most of us will have memory Th17 cells specific for Candida albicans present in our blood. Okay, so this is where where Th17 cells are shown to activate secondary neutrophil recruitment, and neutrophil phagocytosis is shown here. So here the fungi are labeled green and the neutrophils are labeled red. So once there's a lysis of this fungi, it becomes yellow. Red and green under the confocal microscope appear yellow, and this is what you see. And these are the secondary neutrophils that were recruited by Th17 cells that was reported in this particular study. Okay, so we can, a lot of these research came from mouse models. So we do have a mouse model studying this. So here we infect these mice with fungi. So here what happens first is because of this infection in the local oral cavity, they are unable to eat and they lose weight and they have severe inflammation in the mouth and the gut. And that leads to weight loss. So this is sham, which is the control. They don't lose weight. And the mice that were infected with Candida albicans CA, they lose weight, but because these are immunocompetent, they are fine, and their weight is fine at the end of the 6 days. Okay. However, when you treat these mice with cortisone or any immunosuppressant, they continuously lose weight, and they have to be sacrificed by the day 5 because their mouse is so sick. That basically demonstrates that you need an immune system to actually clear this fungus. So this is actually, evaluating or estimating the number of fungi that's growing on the surface of the tongue using the lysates. So what we have done here is, infected the mice with fungi, and then with immunosuppression, you can see the loads of fungi is really a lot more than the mice that did not get immunosuppression. So here we also can look at the growth of fungus on the surface area of the tongue. So what you're looking at is the tongue epithelium right here. And the papillae structures on the edges of the tongue, and the mice that were infected with Candida, you can see a lot of infiltration of these blue cells. These are H&E staining, so you can see infiltration of immune cells, and they are clearing the infection right here. But with the infection, with the cortisone, you can see these pink structures that are detected by a staining called PAS staining. Which is periodic acid shift staining that actually detects the glycoproteins on this Candida fungus. Okay, so that appears to be pink, and then there's a lot of inflammation going on. But this is an unresolving inflammation because there's not enough immune cells or enough active immune cells to actually clear the infection. So you can see fluffs of hyphae growing on the surface of the tongue. So here we have, so this is infected, uninfected, but sometimes when you infect without an immune system, without Th17 cells, that's what is happening here. Okay, so when you don't have Th17 cells, there's a lot of inflammation going on, but the the fungus is not cleared, okay, it's still sticking to the surface of the epithelium, but there's a lot of inflammation going on. So these basically tells that you need Th17 cells to, and also regulatory T cells, like we also tested regulatory T cells in this model. So both of them are required for elimination of the fungus itself and the elimination of inflammation. Okay, so that's about it, about the fungal, antifungal immune responses. So in spite of it, people get fungal infections. So let's look at it, what the fungal treatments or antifungal treatments are in place as of now. So we have fungistatic agents that inhibits the growth of an organism, and fungicides, it has the ability to kill the organism both in vitro and in vivo. So these are fungistats or fungicides, and most likely or most commonly, a combination of these agents are used to treat fungal infections. And so the problem is there's a lot of antifungal agent resistance going on, which is why people try to go in for more of immunotherapy than using these combinations, because these fungi become very resistant to a lot of antifungal treatments. Yeah.
When we say the organism here, we're referring to the fungus?
Fungus, yeah. So, so we want to avoid the antifungals resistance, and that's why we have to go for more biological therapy, and that's why the research is being done. So some of the common ones that are still that are in clinics are polyenes, such as amphotericin B, nystatin, okay, so we'll look at the mechanism in the next slide. And azoles are most commonly used for Candida infections: imidazole, ketoconazole, fluconazole. But as of now, there's a lot of fungi that are fluconazole resistant, and but azoles are very effective in eliminating fungal infections. And flucytosine and echinocandins are other types of beta-glucan inhibitors and DNA RNA synthesis inhibitors that are also used as a part of the antifungal regimen. Okay, so these are the mechanisms. So here you have a fungal cell, that's the nucleus, and here you have a fungal cell membrane. It's made of proteins, beta-glucans, chitins, and the cellular membrane is made of a lipid-like substance in the membrane called ergosterol. Okay, so these are the ones that are targeted by the antifungal treatments that we just looked at. Okay, so here flucytosine is generally used to inhibit the DNA and the RNA synthesis in fungi, and whereas echinocandins that are recently most commonly used are blocking the beta-glucan synthase enzyme that produces this beta-glucan structure on the surface of the membrane, okay, or the fungal membrane, okay. And here, azoles bind to ergosterol and inhibit this, so the fungal membranes are incapacitated, and so imidazoles target the ergosterol part or the cellular membrane part of the fungi. So these are the sites of action where current antifungal treatments are working. So with that, we end the lecture, and this is for further reading, and that's basically the syllabus as well. And this is where I got the slides from. But if you're interested in more details in any of these, you can go through this and